Hello students, in this section we will study about different components of enzyme as well as the factor which affect the enzyme. Firstly, we have seen that mostly enzymes are made up of specific and tertiary protein. It means we can say that mostly enzymes are made up of protein. It means components of enzyme. Enzymes are made up of specific or tertiary protein. So we can see that enzymes are made up of protein. The main components of enzyme is protein. On the basis of component, we categorize enzymes into two different categories. On the basis of component, there are two types of enzymes. Which one? Simple enzyme and conjugated enzyme. Simple enzyme and conjugated enzyme. If an enzyme contains only protein, there is no other substance may be present, only protein contained, that enzyme is called simple enzyme. The enzyme which contain only protein, no other substrate or no other substance is added, that type of enzyme is called simple enzyme. For example, pepsin and trypsin. These are the enzymes which contain only protein, so they can be categorized into simple enzyme. Conjugated enzyme, the enzyme which contain protein and non-protein part. It means the enzymes which contain protein plus non-protein part. If the enzyme contains protein or non-protein part, that enzyme is called hollow enzyme. For example, if any enzyme contains protein and that the non-protein part is cofactor. It means an enzyme which contains protein plus cofactor, because in this type of enzyme, the protein contains enzyme, only protein contains enzyme is a non-functional. But with the help of cofactor, this enzyme becomes functional or activate. This is called hollow enzyme functional. It means when enzyme contain protein plus cofactor. For example, this type of enzyme is conjugated enzyme is apoenzyme plus cofactor, because only apoenzyme is non-functional, but as the apoenzyme gain cofactor, it becomes functional. At this time we can say that hollow enzyme functional. These cofactors which are non-protein part, they may be categorized on two different types, because these are non-protein part. These cofactors are of two types. Which one? Organic non-protein. It means they have protein and non-protein part, but they are organic. And second one is metallic ions. When metallic ions, the cofactors are metallic ions, react with non-functional protein, non-functional enzyme, they always activate. So metallic ions cofactor are known as activators. Activators. Organic non-protein. It means they have non-protein part, but they are organic. These non-protein part separated from this type of enzyme, because apoenzyme is non-functional. But as the apoenzyme get cofactors such like as organic non-protein, it becomes functional. But non-protein part may be separated from apoenzyme or cannot be separated. So we can say that organic non-protein is also two types on the basis of separation. On the basis of separation, it means, what do we mean by separation? These non-protein part, if separated from this non-functional enzyme or not separated, easily separated. If it is easily separated or cannot be separated, we categorize these organic non-protein part on the basis of separation two types: easily separated or cannot be separated. Example of easily separated is coenzyme, and cannot be separated easily with non-functional enzyme, that is prosthetic groups. So we have seen that these are different components of an enzyme. On the basis of function in the cell, there are two types of enzyme. It means we can categorize the enzyme on the basis of function in the cell. Those two types are, the enzymes are of two types on the basis of function in the cell, which are endogenous or exogenous. These endogenous enzymes are also called intracellular enzymes. And second one is exogenous, or we can say that extracellular enzymes. The enzymes which function inside the cell, those enzymes which function inside the cells are called endogenous or intracellular. It means they function inside the cell. In prokaryote, all the functions are performed inside the cavity or cell. So we can say that these are endogenous enzymes. Exogenous or extracellular, the enzyme which doesn't function inside the cell or cavity but outside, such like as in lumen, those are exogenous or extracellular. It means outside the cell, means cell cavity. Those are called exogenous or extracellular enzyme. These are the different components of an enzyme. And the next one is the factors which affect the enzyme. Factors which affect the enzyme. There are many factors which affect the enzymes, because in these factors, in presence of these factors, the enzymes show more functional and some may not. First of all, the factor is temperature. all enzyme functional. The range of the temperature 25 degrees Celsius to 40 degrees Celsius. It means temperature range is 25 degrees Celsius to 40 degrees Celsius. In this range of temperature, all enzymes show functions. All enzymes show functions. We categorize this temperature in three different conditions, such like as minimum, optimum, or maximum. If the temperature remains lower than 25 degrees Celsius, then the function of enzyme becomes non-functional. It means in this range, lower temperature than 25 degrees Celsius, enzymes show no function, no activate. It means enzymes are no activate in this range of temperature if the temperature remains 25 degrees Celsius. Optimum, if the temperature remains between the 25 degrees Celsius to 40 degrees Celsius. As the temperature starts from 25 to 40, the enzymes show more activate, enzymes show more functions, and that is called optimum condition or optimum temperature. In this range, the enzyme are activate and show more function. So this is called optimum temperature, temperature optima. This is called temperature optima. What is temperature optima? The temperature at which the enzyme show more and more functions, because at this temperature the enzyme becomes more active. So this is called temperature optima. Maximum temperature. If the temperature beyond exceed more than 40 degrees Celsius, we know that the enzymes are made up of proteins, and in high temperature these proteins may break down its bond, and if the proteins broke down, then the enzymes will also denaturate it. So at high temperature, enzymes denaturation or denaturated. It means breakdown of bond between the poly enzymes जो भी contain होते हैं. Polyproteins, for example, we can say that tertiary proteins, the hydrogen bonds may be break down. So if the temperature remains lower than the activate, then that there is no activation can be take place in enzyme, so there is no function can be shown by the enzyme. Optimum temperature, if the range remains 25 degrees Celsius to 40 degrees Celsius, at this time, at this range, enzymes remains more active, and more active enzymes show more function. But if the temperature exceed beyond than 40 degrees Celsius, the enzyme denaturation. So we can say that at two ranges, minimum and maximum, there is no functions can be shown by the enzymes. Clear? The temperature at which the enzymes show more function, that is called temperature optima. But in one more exceptional case, as we have seen that more than 40 degrees Celsius, it means all the protein enzymes will be denaturated. But one example is thermus aquatica. It means the bacteria which are thermophilus, they can be survive at 80 degrees Celsius. How they can be survive? Because in prokaryotic cell there is some special conditions in which special proteins which can be survive at high temperature, and those type of proteins may be called as Taq polymers. Thermus aquatica, or we can say that thermophilus archaebacteria, These are prokaryotic. They have special tag polymers which can be survive at more than 80 degrees Celsius. They survive at high temperature more than 80 degree Celsius. This is because they have tag polymer or tag polymerase. Tag polymerase, we will study 1+2 class. DNA polymerase and PCR, polymerase chain reactions. Those prokaryotic organisms contain tag polymerase. Due to this tag polymerase, they can survive more than 80 degrees Celsius. On the behalf of these functions on temperature, we can show that firstly, the enzyme, as the temperature increase, the activity or functions are also firstly increased by the enzyme. But as temperature may increase continuously, it becomes down. So we can say that the increasing of temperature, firstly the increasing of functions rate or functions of enzyme. But if temperature continuously increasing, then the functions stop, because at high temperature the enzymes denaturated. And second factor is pH. Second factor is pH, potence of hydrogen. It means if a solution contains hydrogen concentration, that is called pH. Mostly all the enzyme shows the similar range of temperature, but the enzyme have different pH to show different functions. There are different enzymes such like as salivary amylase. Salivary amylase enzyme which is present in saliva, in mouth, and this contain pH is 6.6 to 6.8, or we can say that only 6.8. It has only 6.8 pH. That is slightly acidic, because at 7 that may be neutral, and salivary amylase contain pH 6.8, so we can say slightly acidic. Pepsin enzyme found in stomach, and in stomach HCl acid is present. Due to presence of HCl, the pepsin becomes more acidic, and pH of pepsin is 1.2 to 1.8. This is more acidic. Trypsin is secreted by pancreas, but it shows function in the intestinal gut, and in intestinal there is alkaline nature. So trypsin in intestinal, it contains pH 8.6 to 8.8. It is slightly acidic. Which one? Salivary amylase. And trypsin is slightly basic. So on behalf of this different range of pH, we can say that different enzymes show function at different pH. Different functions performed by different enzymes. It means the pH of different range is different, but in temperature, there is all enzyme may be function between this range. Clear? And the next factor is concentration of substrate. This is the second. First one is temperature, second pH, and third one is concentration of substrate. Suppose we have an enzyme. This is an enzyme, and it may contains 1000 active site. It contains 1000 active site, and we have only 500 substrate. All the 500 substrate attached to the 1000 active site, because there are 1000 active sites at the enzyme. So all substrate may be combined and product will produce 500 product. If there are 800 substrate, they may also bind with the 1000, because there are 1000 active site. So product will be produced 800. If there are 1000 substrate and there are 1000 substrate and 1000 active site, all substrate may be bind with the enzyme, then the product will also produce 1000. Is the similar? But when the substrate amount is exceed more than 1000, it means if there are 1100 substrate, out of 1100 substrate, only 1000 substrate may be able to attach with the 1000 active site. The rest remains, 100 substrate remains without any production of product. So the product may be always produce 1000, not 1100, because there is only 1000 active site. So we can say that if firstly the substrate amount is increased, the function of enzyme is also increased, but up to a limit. Up to a limit means the enzyme which contain how many active site. It depends on active site of an enzyme. So we can say that the firstly increasing of substrate, increasing the rate of function, but up to a limit. What is limit? How many active sites are present on the enzyme? If the substrate beyond exceed, then the active site, then all the exceed substrate cannot be able to produce product. We can represent it by a graphical representation. On x-axis we can show that the production rate, and y-axis we can show the velocity. It means how much the pH binding site or active site may be present on enzyme. This is called velocity V. Firstly, if there are we can represent 1, 0, 1, 2, 3, 4, 5, 6. It means this is one line number. Firstly, the rate of reaction as increase the substrate. The rate of reaction is also increased, but up to a limit. It means this is the limit. For example, here is the 1000 binding site may present and 1000 substrate is attached. This is the maximum point at which the production may be take place. And from here we can join this, and this higher production is known as Vmax. Vmax means maximum velocity can be shown by the substrate. Clear? The very important question is, many times in NEET, this question is asked, what is mechanism constant? What is Michaelis constant? Michaelis constant is always the half of Vmax. The maximum production can be take place if we make the half. What do you mean by half? Suppose it is the main peak point at which the production can be take place. If this is the half, and from here we can say that this is the half of Vmax. So it is 1 by 2 Vmax. And 1 by 2 Vmax, V stands for velocity of maximum. That will remains half. This is called Michaelis constant. And it is represented by Km. One more example we can take is this. If there are only other substrate which may be added to any substance and the substrate may be reached here, this is the maximum velocity of the production of this other substrate or other substance. This is the Vmax of other substance. Suppose this is the S1, substrate S1, and this is substrate 2. The Vmax of S2 is this one. And if we want to make it a Michaelis constant, it means Km. What is the Km? It means the half of this one. Half of this one, it means here. If substrate S, the 1 by 2 Vmax, it means Michaelis constant is equal to 1. This is the 1 of substrate S. S1 and S2, the 1 by 2 Vmax is nearly 0.7. Nearly 0.7. Here is the main question. The substrate which have lower Michaelis constant, that has higher affinity with enzyme. Lower Km Michaelis constant, the higher affinity shown by the substrate of an enzyme. Clear? And these are the different factors which affect the enzyme.